this is Dr. Krishnan Ramanathan. As I mentioned earlier, my classmate from in lower primary school. And he has visited Sahrdaya in 2012. And now he's sitting from Basel in the head office of NOVARTIS. So interestingly, it's very nice to hear about Krishnan's background, because he's a mechanical engineer from IIT Chennai. And later on, he moved to US to do his master's in biomedical engineering, and then later PhD in biotechnology engineering. And now, after that, he has been working with Novartis. I think all over the globe you have moved up, isn't it, Krish? Except Australia, I believe. So he has been in US for quite a long time, and after that he moved to Europe and in Basel. And then he was sent to Russia for 2 years as the chief scientific officer there, and he established a center, the Novartis Pharma's presence all over Russia. So he's been very efficient in that. And right now he's back in Basel, and he's the chief senior global research officer, right, of Novartis. A really big shot whom our classmates are really proud of, the topmost in our class. So welcome, Krish, to this faculty development program. Over to you.
Thank you, Amity. Thank you for the kind introduction. So it's always a pleasure to see you. And welcome to the faculty development program to all those who have made the time to attend this 2-week session. I'm very delighted and privileged to address this group. I already mentioned to Ambali that I'm not considering myself an academician anymore. I'm in the industry side, and I'm on the side where we are trying to implement some of these things, that ideas and innovations that are developed at academic institutions. So please pardon me if my talk is very general and not, giving you some techniques that you may be thinking that you will pick up over the next many days that you're attending this faculty development program. So I thought I would focus my talk today not only on the technology side of how to develop biopharmaceuticals, but to look at the economics. And I've been made, in some ways, acutely aware of this, because I've traveled the globe, as you've seen in some of the short introductions that Ambali has made. And medicine access in different countries is very different. So medicine access in India is very different from the access in the United States. And Russia is somewhere in the middle. I think there is an Indian type of access scheme and an American access scheme within Russia. But including in India, it is very diverse who has access to pay for medicines. So I think biopharma makes it relevant because some of the latest medicines that we are developing are actually biologicals. They're not chemicals any longer. So I'll try to give a general talk that gives you some background on what it takes to develop an ecosystem of innovation in biopharma, and why some countries may be successful, some not, why some areas are successful. So to give you some background on this. So I know I've prepared some material and sent, but I think the slide viewer is not available. So I'll just, you have the pleasure of just seeing me in those slides for this session. So the first thing that I wanted to tell you is that over the last 40 years or so, the availability of pharmaceuticals, and especially biopharmaceuticals, has vastly improved the life expectancy of people. Life expectancy and also the quality of life. And why is this important? Because in some ways, this is what justifies where we spend our monies and where we spend our public monies, our private monies. I think the age-old search for longevity is starting to bear fruit. If you just look at the history of mankind over the thousands of years that we've populated the planet, our life expectancy went from probably 35 years in prehistoric times to maybe 65 years. So we added, over thousands of years, we probably added, I would say, maybe 20 years, 30 years. But over the last 40 years, we've added quite a bit. In many countries, this has gone up by 20 years. And imagine this short period of last 40 years in the history of mankind. Why have we added so many years to our life expectancy? There are two reasons. One is we have reduced the number of people, number of newborns who are passing away. And most of this is from infectious disease. And we've been able to curtail that based on the immunizations we provide to children And making these little young babies live longer adds a lot to the life expectancy, because it's a bit like you have a bell curve of how long you can live, and if you take out that tail of very young deaths, then you add a lot to the mean. It moves to the right. The second reason why this life expectancy is increased is because of medicines. And medicines have made us live longer with cardiovascular illnesses, if you will, or actually make our heart pump longer over periods of time, when you can treat yourself with basic conditions like hypertension, heart failure, are very treatable today. And it's very rare that in the developing world you would die because of hypertension. You would die because your heart's failing and you extracted the last amount of pumping effectiveness that it can do. But this life expectancy gain comes at a cost, right? And there is a very interesting plot that is developed by many of the health economics institutions of countries that has a clear correlation of GDP production in a country to life expectancy. So India falls somewhere in the middle. On the very low end, you have countries that have very poor economic conditions, even worse per capita GDP. And then on the the high side, you have small but effective economies that have about the 10 times per capita production per person in GDP. And they have about 10 years average increase in life expectancy from the median. So this cost of economics of what actually goes into a health system decides how long you live. And that's why I think investing into biopharmaceuticals is important, because it allows people to live a longer and a more fulfilled life, with better quality. And when I was researching for my talk, I must admit, this is not my core area now. I'm working more on the R&D, trying to develop new medicines, trying to figure out which projects we invest in, which projects actually people are willing to pay for. I'm a little distant from the technology side, but still, I think I'm close to it in terms of reading up and making sure that we invest in the right technologies. And as I was researching this, one of the things that I noticed is that one of the most simple things that adds to longevity or life expectancy is having access to clean water, right? And we often forget how important this is, having access to clean water in the large scheme of things. So if you look at, again, the different countries that I mentioned that have long expectancies of life, the basic amenities are almost guaranteed. And in countries where the basic amenities like water are not guaranteed, you have a lower life expectancy. And this results in also an expanded situation when you think about biopharmaceuticals and access. Now, coming back to the biopharmaceuticals and even the terminology of why we call it biopharmaceuticals. If you look back into the history of pharmaceutical companies, most of the pharmaceutical companies were chemical companies. Novartis today is formed from a merger of two big companies. One was called Sandoz. In India, you might have these tablets, calcium Sandoz, that you feed to children to give them bone strength. And this is one of the staple products of Sandoz. And another company is called Ciba. And Ciba Geigy, you may be familiar with, again, in India is a fairly large pharmaceutical company. And these were historically chemicals producers. They made chemicals that helped make dyes for fabrics, that helped paint for walls and paint for different colors. And there were chemists who were researching how to produce different synthetic compounds to give you colors. Even before many years when, during the Silk Road times, silk used to come from eastern part of the world to the west, and the west used to put color into these synthetic colors because there were very few natural dyes that was available to make different color patterns in fabric. Yeah, you have to take these dyes from naturally occurring color sources like flowers. And you don't have access to many of these in the Western countries or in the non-tropical areas. So they researched into chemicals to find out, how you can synthesize artificial colors. And many of the pharmaceuticals are actually accidental discoveries of these chemists that seem to work in large plants and seem to think, I seem to be breathing better. Is it something that I inhaled from the chemicals? And maybe there is a cure for asthma over there. Or you accidentally ingest some of these chemicals and it lowers your blood pressure. It takes down your load of vasopressin or something like that. So these accidental discoveries became medicines. But that paradigm has changed. Today, since 1990s, we've sequenced the human genome. So what does that mean? We know what's the code behind developing humans or developing organisms. But simply access to the code is not enough. Over the last tens of years, we've even figured out what does code mean? What are these genetic code making? What are the proteins, which are the building blocks of life? And most of pharmaceutical R&D today is in trying to identify proteins that are amenable to turning on and turning off. So these are basically enzymes that you're trying to figure out how you can block or accelerate in order to help with certain biological processes. And this is not cheap. You probably are all working in the biotechnology sector and trying to find new techniques. To make a new protein is a lot of work. And the body seems to do it pretty easily. You can feed the cows a lot of grass and they generate milk protein and casein, albumin, and so on. And these are our largest biopharmaceutical entities, if you will. They're roaming around the globe, converting fertilizer and grass into milk protein. But to make it synthetically is a challenge. To get a bioreactor in place, to develop proteins that are actually therapeutic targets are extremely expensive. So if you look at an industry overall, pharmaceutical industry spends about 18 to 20% of our sales into research and development. I mean, can you think of any other industry that spends so much in R&D? And I have a list here. I think the next comes the semiconductor industry, which is all the biotech chips that you're looking at today, and they spend about 14%. And if you think that aerospace or technology, flight, are expensive proposition, these companies only spend about 6% into research and development. So investing into healthcare is expensive, and of course the payback is good. You live longer, right? You have proteins that can help you live longer. And why is this expensive? Why is developing biopharmaceuticals expensive? I mean, there are two aspects of it. One is the production aspect. The producing these biopharmaceuticals are expensive. They take a lot of investment into new technologies, and these require a lot of capital investments. The second part is that there's still a little bit of serendipity in drug discovery. So the drug discovery and development goes through many phases. Initially it is identifying a target, identifying a protein or an enzymatic pathway that you can inhibit or open up, and these pathways can be triggered by different kinds of proteins, different antibodies, different chemicals. And often when you test them in the clinic and in preclinical stages, these things turn out to be either not so good or they turn out to be toxic. So there is a success rate that is built into the development of new medicines. And out of 10 medicines that we test in humans in the form of phase 1 clinical trials, there's only one that makes it through. I think nowadays with biopharmaceuticals, this success rate is becoming close to double. There's close to 2 molecules out of every 10 if you just take biopharma. But if you take the chemicals itself, it's less than 1. Because many of these turn out to be toxic. Either they cause a liver toxicity, you're not able to metabolize them in the liver, you're not able to excrete them through the kidneys. And these chemicals are essentially foreign entities, and that's why they turn out to be toxic. And biological proteins, or on the other hand, antibodies, for example, their toxicity is lower because proteins can be degraded without having to overexpress liver enzymes. And most of the toxicities coming from these proteins are what we call as on-target issues. They're not off-target. And if you have a medicine for hypertension, it could cause liver problems, and that's off-target. But if you have a medicine for B-cell therapies, the issues that you usually find in proteins is that the effect is from reducing B-cells by a large degree, and so it is on-target. And that's one of the benefits of biopharmaceuticals, is because you can almost picture the toxicity and you can figure out the right dose to give. Because there's some of the most senior drug developers, they say that drug development is an art and everything depends on the dose. High doses, it's a toxin. Low doses, it's a placebo. So you need to find that. And with proteins, it's a bit easier because you have a big therapeutic window with which you can dose them. Higher doses don't turn out to be as much toxic, and you can even find an efficacy at small doses of this. So I'm going to give you one disease example that I'm currently working on to sort of highlight what biopharmaceuticals have done. So the disease that I'm spending a lot of time today is called multiple sclerosis. It's not very commonly diagnosed in many of the tropical areas. People, in fact, don't even know how the trigger happens. So I'll tell you a little bit about the disease. So the disease comes from an autoimmune attack on the nerve cells, on the neurons. And the autoimmune attack is a trigger or a breakdown of body's tolerance where your T cells are going and targeting your neurons and chewing off the myelin sheath, which reduces the conductivity. So the first manifestation of symptoms usually come in the form of muscle weakness or vision loss, where your optic fibers or the neurons going from your eyes to the brain have reduced their conductance, so you're not able to see very well. And what happens with these patients? They deteriorate fairly rapidly if you don't treat, and you may have acute vision loss leading to blindness. You can have disability, unable to walk from the muscular weakness resulting from demyelinated neurons. So historically, the first treatment for this was treating them with interferons. And then the effect of interferons was basically to trigger the immune system to act against our own autoimmune mechanisms. And these are challenging. And the efficacy of this was low. Today, over the last 30 years of drug development, we have small molecules, chemicals, some developed by Novartis, many developed by other biopharmaceuticals. And of late, there have been biological products that are B-cell depleting proteins or antibodies that have a profound efficacy in reducing the antigen presentation of the body and giving these patients a much better quality of life. Now, this has resulted from tens of years of research trying to understand the etiology of the disease, which we still don't fully understand, but we are able to break the pathway by which the disease manifests. So if you're a 25, 30-year-old person who was diagnosed with multiple sclerosis, 10 years ago, 15 years ago, pretty much they would tell you that in 15, 20 years, you would be in a wheelchair, right? Because you will lose your muscle weakness. We don't have so many patients in wheelchairs now because 10, 15 years ago, they started getting treated with many of these therapies that are called disease modifying. And this has come with millions of research into investment in biotechnology compounds and biopharma, and that's what's made us help these patients. And this, in some ways, is what keeps me going, waking up every morning and coming up saying that we will come up with a better medicine that will help them live longer. And I use this to illustrate what has happened in the world of biopharma because we've got about 30 years of development during which time new medicines have really added value. So I've looked into what has the FDA, the US Food and Drug Administration, approved in terms of biotech compounds over the last 30 years. And this is a big growth curve. So I think the FDA itself has been studying the biotechnology space and has been approving more compounds that are biopharmaceuticals. And I just picked up the top 10 compounds that are actually biopharmaceutical entities. The top-selling biopharma compound today is Humira, adalimumab. It's not made by Novartis, it's made by AbbVie. And this was, I think, 15 years ago when it was first approved. And it's a inhibitor of TNF-alpha, which gives an anti-inflammatory mechanism that helps inhibit pain from rheumatoid arthritis. It also helps in multiple other diseases that are triggered by breakdown of autoimmune conditions. Psoriasis, for example, Crohn's disease. And this, per treatment today, costs over $25,000 US for a year in the United States. It's probably a bit cheaper in India and other countries, but not by that much. If you buy Humira, it probably costs at least $10,000 US for a year of treatment. And that's expensive. If you think about who can afford to pay that to treat their own rheumatoid arthritis, very few, I would say. Maybe 2% in the world. And this makes it a challenge for discovering new medicines, because who are you trying to help at the end? You're trying to make it accessible to all patients. And that's where I think expertise like yourself, who are learning new techniques, can come in that can help make biopharma accessible for all patients. We need to find technologies that can make this like aspirin, that can make this like paracetamol or ibuprofen, that can be bought at the pharmacy at a much cheaper price. And I think we will get there. It will take us a few more years. But this is, I think, one of the biggest challenges for the biopharma industry. I picked up a few other compounds. Another one that comes up is called rituximab, Rituxan, which is an inhibitor of CD20 that is expressed on B cells in the immune system. And rituximab is a very interesting compound. It was first researched in cancer, where there are many B cell lymphomas that lead to proliferation of B cells. And through our research, we find out that many of these B cells have a marker protein, which is a CD20, is a common marker that is expressed on B cells. And if you can make an antibody that can bind to CD20, you somehow make these B cells themselves a target of apoptosis or of lysis. And thereby you can deplete these B cells. So this became very popular in treating B cell cancers, where you can stop the B cells from dividing by making an antibody to a protein expressed on the B cells. But over the last tens of years, this therapy has been used in multiple other conditions. And most of these conditions are a breakdown of the immune system's tolerance mechanism, where your B cells start expressing antigens that are basically proteins in your body. And you develop rheumatoid arthritis, you develop multiple sclerosis, you develop psoriasis, ankylosing spondylitis. Many of these autoimmune conditions that are a result of your immune system attacking your body's proteins can be stopped by blocking CD20 expressing B cells. And so rituximab has become extremely popular. One of the challenges of rituximab is it's a chimeric protein. And if you understand protein biology, this antibody was expressed or first identified in mouse. And then a chimera, a compound of mouse and human proteins, was created, by which it made it more manufacturable, easily discoverable. Now, most of the new antibodies that we are working on are human antibodies. And there is a very interesting technology that helps you develop human antibodies, and these are done in mice which basically have a human immune system or express human B cells and T cells. So by presenting an antigen to these mice, you actually generate antibodies that have human sequences, genomic sequences, and thereby they're less immunogenic. And there are multiple other compounds. Another popular biotech compound is called Avastin. It's an inhibitor of growth factor, the VEGF it's called. And this one is again used in multiple cancers. Most of these cancers have a mechanism of providing blood and nutrients for themselves. And thereby, if you cut off the nutrient supply, the growth of new blood vessels to treat these cancers, you can stop these cancers from growing. And that's the mechanism of Avastin. Novartis has developed another molecule that's a sort of a child of Avastin, which is a VEGF inhibitor that can be injected into the eye. And injecting these into the eye can reduce things like age-associated wet macular degeneration. And the macula of the eye degenerates because there are new blood vessels that come up from behind the eye and they push off, and you can even have a retinal detachment. And by injecting these into the eye, you can actually save a lot of people from blindness. And this has been possible over the last 15 years because of our research into the pathway and understanding the biology of some of these diseases. Another core biotech compound is a vaccine which is called Prevnar. It's made from a polysaccharide conjugate expressed in many bacteria, pneumococcal bacteria. And this is extremely important in the developing world, where preventing these infections in children reduces the amount of disease in the community. Now, I have a little bit of time, it looks like, so I'm just going to look at biopharma production and why it is so expensive. And from my understanding, my somewhat simplistic understanding, you're looking at an upstream process and a downstream process of production. And the upstream process involves, figuring out how to clone and how to make these proteins that can be made as recombinant molecules into a cell line which is easy to propagate. So the cell lines where we would like to generate most of the new antibodies would be either mammalian cell lines or human cell lines, because producing antibodies in a condition that is as close to the condition as the human body is important in terms of the post-translation post-translation mechanisms like glycosylation, etc., which happens better in mammalian cells. Now, the upstream process is extremely expensive, and it requires a lot of focus on quality. Creating the right banks, the right cell banks with the right plasmids that have the right structures that can be propagated so you have less genetic drift. Harvesting these at the right time and expanding them through serial fermentation steps, going to a small scale, to a medium scale, to large fermenters, can be thousands of liters. And many of the times, this quality-prone process is, you can see the cost of this if you have to throw away a large batch of fermentation that has had bacteria growing in it, for example. Because these things are growing in media that is rich for growth medium, for growth of cells, and quality and focus on sterile environments is really important in the upstream process. And making sure that the protein is repeatable, that is reproducible, the same protein that you test and assay when you first started the manufacturing process 10 years later is extremely important. And these in chemical technologies are much easier, but in biopharmaceuticals, this becomes a challenge when you see a lot of drift. So the upstream production process is heavy on quality and making sure that you have the right banks and producing a fermentation process that yields a lot of protein. And then this protein needs to be extracted. And even today, with a lot of technologies available, we still go through chromatography is our best approach for extracting this protein. And as many of you know, chromatography is separation of molecules based on their size. And this process at best is quite crude because most of the proteins are about the same size. So if you want to extract a certain volume of protein based on its molecular size, you will get a lot of ingredients that are coming up along with it that are the same size in terms of the molecule, but they're not required. They're not the protein that you want. You can improve the purity by using affinity chromatography, by separating them first based on a cation or an anion exchange, and this is based on the charge of the protein, because if you know what is the charge structure of your protein, you can create an affinity column that can provide a window where you can target your protein and elute it at the right charge. There are further techniques that are coming up today where you can even make a column that's based on an antibody to your own protein, so you can then discharge it. If you can do it in a reversible way, you can make a more pure sample. Most of these production processes require heavy scaling and investment in quality, and they're expensive. And that's one of the reasons I think having faculty like you try and figure out what are the best ways to make these affordable to people across the globe. And I looked at the manufacturing plants in the globe today, and many of them are in the United States and Europe. There are a few coming up in India and in China and in countries where there's high demand for biopharmaceuticals. But we still go a long way from having a chemical plant that can produce tons of aspirin, for example. So I want to close my talk with sort of 3 points that I think are really important for you as faculty to understand in what triggers biopharmaceutical innovation. The first is knowledge or know-how. And this is part of the education that you're coming in, is trying to understand how to make new biopharmaceutical. And this requires an integrated process from high school to college to academia with research and industry to make sure that you can translate your learnings from college and academia very quickly in the industry, and reverse translate as well. Learn from industry and what needs to be scaled and produce it back. The second one is funding, how you make investments in this. And this funding can come from private money, from companies like Novartis. Funding can come from the government, public monies, from taxpayer money. And it can also come from individuals, from choices we make in buying medicines. in whether we buy the generic medicines or invest in those that have invested the time into R&D, focusing on the brands that provide you the quality. And the third one, which is really important, is regulation. And the government plays a large role in regulating these, because regulating them means you provide the best safety